All right, so now we're going to move on to our fourth presenter for today, Dr. Anthony Pattison from Department of Agriculture and Fisheries from Queensland, Australia. So Dr. Anthony will be presenting his work on the role of non-banana plants in Banana Fusarium Wilt management. The floor is yours, Dr. Pattison. Do we have Dr. Pattison in the room?
We do have. Dr. Anthony has to unmute the microphone.
Dr. Anthony, could you do that?
That's unmuted. You can hear me now.
Yeah, cool.
Yeah, sorry. There's just a...
the floor is yours, yeah? Whenever you're ready.
So you can see my screen now?
Yeah, I can see your screen now. You can put it in the slideshow mode, yeah?
It's going into slideshow. Okay, well, thank you very much for the introduction, and thank you very much for the opportunity to speak. As introduced, I'm Tony Pattison, and I work on bananas, and I'm presenting the work of a large number of people from a large number of institutions on the role of non-banana plants in Fusarium wilt management. So just to give you an overview of what I'm talking about, I want to just wanted to try and give some context of why I'm looking at Banana Fusarium wilt at non-banana plants, and then look at alternative hosts, non-banana plants as alternative hosts, before moving on to rotation crops, and then looking at non-banana plants as cover crops and intercrops. So then by the end of the presentation, I hope you've defined that non-banana plants can be either desirable, that is, they reduce disease, or they're undesirable and they sustain Banana Fusarium wilt within the plantations. So as we've already heard, Fusarium tropical race 4 is spreading around the world. There are now 22 countries where the disease has been found, and most recently, 5 new farms have been located within Peru in South America. And we've also heard about how it infects the plants. That is, it moves into the roots through the soil, into the roots of the plant. It moves into the xylem tissue, into the rhizome, before moving into the pseudostem and blocking the pseudostem, causing the plant to die and senesce. Once the plant senesces, the fungus moves out of that xylem tissue, forms chlamydospores, and as the plant decomposes, those chlamydospores are returned to the soil, where it creates a new infection, infecting more roots. And this has been summed up very nicely by a paper by Nolene Warman and Liz Aitken. But as well as how the disease goes, we need to think about how bananas are produced in a commercial banana production system. We often see infected bananas. We'll knock out that crop. Farmers will remove that crop, have a fallow period for a certain period of time before they replant bananas, and hope to reduce the disease and have a continuously returning system where they can market bananas. They also hope within that system that they're increasing the number of antagonistic organisms, the beneficial ones, to help suppress the disease. But non-banana plants can either be alternative hosts and carry over the disease. We can have rotation crops as farmers try to utilize the land and try to grow something else to make money while it's out of banana production. Or we can have ground covers or intercrops in amongst the banana plants. So let's start off with the alternative host story. So what we did in Australia is we looked at the main banana production areas, firstly in northern New South Wales, where we went out and sampled fields with alternative hosts to race 1 and subtropical race 4 in northern New South Wales. In North Queensland, we did the same for race 1. This is where 90%, 95% of the Australia's bananas are produced. And we also did the same in the Northern Territory, where tropical race 4 is endemic. So the results from the field surveys in northern New South Wales of 46 different weed species were found, 6 collected were found to be host to race 1. 1 was to subtropical race 4. In North Queensland, 115 samples were collected of 20 different species, plant species, and 4 were found to be host to race 1, the VCG 0124. In the Northern Territory, over 435 samples were collected of 33 weed species, and 6 were found to be alternative hosts to tropical race 4. The main weed species were then taken into the glasshouse, and in northern New South Wales, 8 different species were tested in inoculated studies, and 6 were found to be hosts to race 1, 1 to subtropical race 4, with one species, Ceratro, found to be a non-host. In North Queensland, 50 potential weed hosts were tested, with subtropical race 4 recovered from all of them. And the same story was true for the Northern Territory, where 12 different potential weed hosts were tested. Moving further into understanding what's going on, some of the work that Elizabeth Aiken in her lab with Jay Anderson did was to understand 2 species in particular, cobbler's pea and blackberry nightshade, of how the fungus moves in these as alternative hosts. So they used a green fluorescing protein transformed Fusarium subtropical race 4, and they found that it formed nets around the roots of those plants. It didn't form necessarily into the epidermal cells, but there were nets that could produce chlamydospores. And once those plants were treated with herbicides, then there became extensive hyphal colonization, and then sporodokia produced on the outsides of the plants with massive chlamydospore production. So all the information that we've been generating on alternative hosts, we've put into a guide for our banana growers in Australia. And so this has photos of the different alternative hosts, the names, locations where they're found within Australia, description, and then a glasshouse rating to Fusarium and a field rating, whether it has been found in the field. So I've presented a lot of information on alternative hosts. So I just wanted to sum up before moving on. All plants are potentially alternative hosts to FOC. The host status depends on the inoculum level in the soil, and that some alternative hosts are better than others. It's difficult to isolate Fusarium oxysporum from the roots of alternative hosts in the field. But once plants are senesced, if we're using herbicides or killing them, then they're extensively colonized by the saprophytic ability of Fusarium oxysporum cubense. So moving on to the next role of non-banana plants, and that is as rotation crops. So the way that we've been screening these in Australia is to do what we call a pot-in-pot experiment, where we have a pot inside another pot, and in between is filled with soil and our test species. We grow the seed, and 2 weeks later, this is inoculated with an FOC isolate. The crops are then grown for 12 weeks before they're harvested, and then a root rating done and recovery of Fusarium oxysporum. 20% of the tops are then returned to the tops of the pots, and a tissue culture banana placed in the middle of the plot once that central pot has been removed. This is then grown for another 12 weeks before a disease assessment is done on the plants and a destructive harvests. So I'd just like to go over some of the earlier results, recent results from some vegetable crops that we've been looking at. In this, we're looking at 4 different vegetable crop species: beans, chili, corn, and tomatoes, and looking at 3 different cultivars of each. So at the end of that rotation crop phase, there was beans and corn tended to have more Fusarium recovered from the roots than either chili or tomato, and there wasn't a lot of difference between the different cultivars. However, once we grew the bananas in the pots and grow them for 12 weeks and then assess the roots, we found a different story. The inoculated banana, which is on the left of the screen, was our control, and most crops had very similar recovery of the disease to our inoculated control. Only 2 species, chili, purple tiger, and tomato tropic, had recovery of Fusarium oxysporum similar to the uninoculated controls. So this suggests that there are differences within the varieties of the different vegetables on how they host Fusarium. Well, this is really important for small growers in Southeast Asia. In Australia, we're more likely to use rotation crops, using pasture species as rotation crops and incorporate them as organic green manures. So some work Wayne O'Neill did, one of my colleagues, was to compare legumes and grasses and different species and cultivars to bananas. He did this over a number of experiments. So the red ticks above the columns that you see on your screen are those lines that have been tested multiple times. So on the blue bar on the left is the susceptible control, and anything that's orange is not significantly different from that susceptible control. So most of the legumes had similar ability to host the disease as banana. However, those green bars are those that were significantly better than the inoculated controls and had similar quorum rating as the bare uninoculated control. So there we see three legume species, but 5 grasses that have potential resistance or usable resistance to Fusarium. In a field experiment done in the Northern Territory by Charles Mintoff and Lucy Tran Wynn and her team, they looked at infected bananas over a number of seasons through a whole cropping cycle. They started off with an infected crop and they measured the amount of TR4 DNA in the soil through the whole cropping cycle. So they grew rotation crops, and in this case, there were 4 different treatments: the fallow treatment, which is a weedy fallow; cavalcade, which is a variety of centrosema; jarrah grass, which is a variety of a digitaria cultivar; and sugar graze, which is a forage sorghum cultivar. So that after they were grown, then the banana crop was replanted into the same fields. And overall, there wasn't a big difference between either cultivar or time in the amount of DNA that was found in the soil. However, when the bananas were grown back into the soil, disease TR4 was starting to be seen 15 weeks after planting in the weedy fallow. And that increased up until the end of the experiment at about 32 weeks after planting. However, with the cavalcade centrosema variety, this was greatly suppressed. The disease symptoms weren't being expressed as highly as they were in the weedy fallow. The opposite was seen with the jarrah grass, where there was greater expression of disease and there was a greater incidence of disease, and the sorghum was somewhere in between. So moving on to my final point now, and that's with ground covers and then growing something in around the bananas, either as an intercrop or a ground cover. So what I'd like to highlight here is an experiment I ran where we looked at different nitrogen and ground covers, which was replicated and ran over 4 years and had 5 cropping cycles. I'll just concentrate on the ground cover side of it. On average, over those 4 years, the total Fusarium abundance in the soil was significantly higher than in the vegetated. That's the red bar on the left, was significantly greater than the green bar where we had vegetated soil. And if we take some soil out of those plots that we had from the field and grew a plant, a susceptible banana plant in there, and then challenged it with Fusarium oxysporum to see if it got disease, we found exactly the same type of result. So those plants that were grown in soil which had no ground cover at all, bare soil, there was more disease than those that were grown in vegetated soil, which had extra plant growth in them. And to understand what's going on here, We collaborated with Paul Dennis and his team in understanding the Fusarium oxysporum microbial subnetwork. So each dot here on the screen represents a species, with the orange dot in the center of the vegetated and bare being Fusarium oxysporum. The blue lines are positive interactions. So as they go up, Fusarium goes up, and the red lines are negative interactions. So as they go up, Fusarium goes down. And we find that in the vegetated soils, 59% of the microbial interactions with Fusarium oxysporum are antagonistic. In the bare soil, there was only 9% of the microbial interactions with Fusarium or oxysporum were antagonistic. So this shows or demonstrates that in the vegetated soil, there is much greater potential for self-regulation of Fusarium oxysporum through those extra interactions with other microbes, helping to regulate that population and impact on the banana plants. So through this presentation, I hope that I've shown that non-banana plants do have a role in Fusarium wilt management, either as alternative crops, rotation crops, or intercropping. And they can either be undesirable, increasing Fusarium oxysporum or leading to a decrease in antagonists, or they can be desirable, such as the ground covers or intercropping, where they tend to decrease the dominance of Fusarium oxysporum and increasing in antagonists. And rotation crops can tend to be either way, and we still need, this is a bigger area that we need to understand on how we can manipulate the populations or the abundance of Fusarium in the soil. So just to reiterate this point, non-banana plants have an important role in the management of Fusarium wilt, be desirable or undesirable. And as alternative hosts, we can tend to think of them that all plants are hosts to FOC. It's just some are better than others. As rotation crops, some plants aid inoculum reduction, encouraging tolerance in the banana crop. And as ground covers or intercrops, they can suppress the dominance of Fusarium, the Fusarium species, and increase the tolerance of the bananas we are growing. So I'd also like to finally acknowledge that this is the work again of, as I said at the beginning, of a lot of people from the Department of Agriculture and Fisheries who I work for, the University of Queensland, and also the Northern Territory Government, with funding from the Australian Centre for International Agricultural Research and Hort Innovation. And we're lucky to have a number of very talented PhD students working on this project to help sort out some of those microbial interactions. And as a last comment, next year we have the Australasian Soil-Borne Disease Symposium in Cairns, of which I'm a convener. And if you're interested, the website's there at the bottom of the screen, and we'd love to see some participation as a hybrid meeting with our colleagues in Southeast Asia. Thank you very much.
Thank you very much, Dr. Pattison. I think we can take one question if there's any, because we have 1 minute left. Or perhaps I can just give you one question. So in your studies, you found 3 types of legumes and also 5 types of grasses that have potential resistant to FOC. Am I right?
That was from the earlier pot experiments. Yes.
Yeah, but when you got into the field, does this resistant effective?
No, this seems to be another mechanisms that's going on as well. So in the glasshouse, we're solely
sorry, I'm losing you.
Sorry, in the glasshouse, we're looking at only the resistance, whereas we're not looking at the potential of beneficial organisms living around the roots. So while we did see some suppression in the glasshouse experiment with some species, yeah, we haven't accounted for that antagonistics, the beneficial organisms that are potentially on the roots, which can increase the tolerance of bananas. And I think that's what we're seeing in the field situation, is not so much controlling the pathogen, but we're getting better tolerance into the bananas through more antagonistic organisms that have grown and lived around the roots of our rotation crops.
All right. Okay, thank you very much, Dr. Tony Pattison. Yeah. So
Yes.
the participant would shoot up questions if they have any for you. So if you could just visit the chat room if there's any for you. So we're going to move on. Thank you for a very, very good and informative presentation just now. Yeah.